I don't know if I told you, but I had an early start. I had an unusual career. Right after high school, I had my own lab. I could do my research the way
Really?
Yes, very unusual. At the age of 18, I had a lab because I was employed as an assistant of a technician, but there was no technician. And the guy who ran the lab, he was interested in teaching and didn't care for lab. So I had a budget. There are good people around I could talk to, but I had very free hands. So at the time, this was early 70s, and at the time there was a form of genetic, in Drosophila we had genetic markers since the 1920s. In humans there were no, only occasional markers, how people can roll their tongue and things like that were markers, but nothing to cover the whole genome. But then came something called isoenzymes, so single amino acid change that can change the isoelectric points of enzymes, and that turned out to be a source of polymorphic markers. A guy called Oliver Smithies, who later got a Nobel Prize for something else, for knockout mice, he had found these isoenzymes in the human, and I just copied what he did and applied it to barley, the plant. Other people around me did. They had 4 markers going, and I put up assays for another 28 and published a paper. I was very proud. But then I went to med school and wanted to do science, and my PhD was much more boring, not very creative, I felt. But then, except I made one invention, which turned out to be commercially valuable, but at the time I didn't think of patenting it. But then in the US, as I mentioned yesterday, the mindset was much more that if it's a good idea, you should patent it and see if it can be commercialized. So when I came back from my postdoc, then that was very much my aim, to develop new technologies and publish them as best I can, but also try to see if they can be put to use outside of academia.
It's such an amazing story.
Yeah, I was very lucky. I had maybe this is an aside, and hopefully I didn't tell you, but I had a letter the other day from a very ambitious first-year civil engineer student who wanted to come to my lab to do an extremely ambitious project, which has nothing to do with what I'm doing. And I was telling him now, well, I'm not sure I can. I have my grants to do certain things. But it struck me that twice in my career I had this complete freedom to do what I pleased. First, right after high school, and then as a postdoc, there were 100 postdocs in the lab I was in. It was a huge lab, very well funded. And my supervisor didn't have a clue what I was doing. So I had complete freedom. I did lots of different things, some of which were quite good, and eventually I started on this ligase-mediated detection, which has been sort of a red thread through much of what I've done. But it was probably a very unusual situation to have this degree of freedom to do what I pleased. Rather frightening freedom, because there was nothing that was more obvious to do. I could do anything or nothing, and I had nothing to build on, so I started from scratch in many directions. And as a consequence, I was very unproductive. I didn't accomplish much for a long time till I finally got something going.
Yeah, but then you get the chance to look into things, I guess, and follow your interests.
Yeah.
And eventually you do find that important thing to pursue.
Yeah, no, in my case, I think it was lucky, and maybe it suited me better than others. But it was really nerve-wracking for a long time, since I was so fundamentally unproductive.
Yeah.
I don't know. There are stories of people who get fired and they don't want to tell their wives, so they go to work, they pack a lunchbox, and then they sit in the park and come home.
Yeah.
I was kind of like that. So I went to, sometimes I was just bicycling around in San Marino thinking, what the hell should I be doing?
Right.
Yeah, I had all the opportunities and nothing was more likely than anything else. It's really scary freedom.
Yeah, because that's one important part of science, right? When you do have that sort of creative freedom.
Yeah.
At some point you have to decide, but then you also need to give yourself enough time.
Yeah.
So how did you cope with that, like with a scare?
I kind of suppressed this, I think. So I didn't really, it only came back to me several years later, the stress I had felt. because afterwards, once things start working, then you sort of idealize and things seem very logical. It's possible afterwards to tell a very logical story. First I did this, then I did that, and it all happened.
That's true.
But it was very random and very chancy, and could easily have gone completely astray. But in the end, I launched on the project. Actually, I had 4 projects I was pursuing, and one of the projects won a Nobel Prize, but not for me, but for somebody else. So it's a really good project, and the other 2 also became major research areas, but I had to drop them because I could only pursue one of them.
Right. Yeah, that's the trouble, right, with choosing.
Yeah.
But you did your postdoc in Caltech.
Yeah.
Can you tell me a little bit about the environment and what that was like?
Yeah, Caltech is a very unusual place. It's almost like a monastery. I think they'd like to see it like that. So there are relatively small research groups that are extremely dedicated to science and do really high level, lots of Nobel Prizes and fundamental research. My lab was not like that. The lab I was in was huge, 100 postdocs or so. And it was kind of frowned upon by Caltech at the time. Also, my supervisor commercialized his research, and he now tells a story how he was called to the president of Caltech and told that this is not really the way we should be working. The commercialism, that's dirty. This is clean science. You shouldn't be doing that. And he also says that. And since then, Caltech has come around 180 degrees. Now it's the other way around.
Right.
Yeah, but so it wasn't typical of Caltech, but Caltech is really a fantastic place with excellent scientists and extremely dedicated people in a very broad range of areas. A very small university. They had 2,000 students at the time.
Right.
I came there as a cellular immunologist, but wanted to transform into molecular geneticist, and that took me quite a while. I had no training. I just read textbooks and tried to get into it. After a while, it can be helpful to enter an area by a different path than others, because you know somewhat different things. Maybe you may not know the stuff that people who took courses know, but then you have a different angle, so maybe you can contribute that way.
Yeah, that's very interesting. And that might also have helped on the innovation journey then.
Yeah, I think so. The great hero, maybe this is an aside, but the great hero of Caltech was Richard Feynman, the physicist, and he tells the story like that also, that he learned mathematics a little different than others. So when there was a problem, if somebody didn't solve it, they talked to their colleagues and maybe they could solve it. But when they have given up, then they would come to him, and he had a different angle, so maybe he could solve it. And everybody thought he was so fantastically talented. It was just that he knew different things. The other one all knew the same thing. So once that was exhausted, then maybe he had a different perspective. In a way, I had a similar perspective on science. I think that I come at it from a different angle.
Yeah, exactly. That's really important, I think. And then to be able also to combine those different perspectives and build on each other.
Yeah.
So that time in Caltech, you told me also that was a very special time in the field, like a lot was happening in molecular genetics. Can you tell us a little bit about that?
Yeah, no, the mid-80s, the lab I was in was one of the pioneering labs behind the Genome Project. They published a paper without I wasn't involved, on automated DNA sequencing. The Signe sequencing was around, but it was automated in a very primitive way, but still demonstrating that things could be scaled. And for other reasons also, people started thinking that maybe we shouldn't go at biology one gene at a time or one disease at a time, but why not learn it all? Why not get the complete overview what the information content in the human being is? And that was one very important inspiration for me, because it meant that it would now become possible to survey all genes and all proteins given sufficiently comprehensive tools. All the assays before were very slow, and you looked at one thing at a time and maybe spent time cloning molecules and eventually finding what you were looking for. Then the other part of this was the PCR, which was also revolutionary. I think it's still, people don't appreciate how revolutionary it is. You search through a very complex genome, 10 billion nucleotides, and in seconds you find 99.9% of all target sequences every cycle. So building tools from synthetic DNA and enzymes, that's also been a pattern of what I've done. So we've been building molecular tools using synthetic DNA and knowledge about enzymes. In my case, the ligases, enzymes that fit things together, have been particularly important, but we've used a broader range of enzymes.
Yeah.
And that still CRISPR-Cas9 is a more modern example of success of synthetic nucleic acids and enzymes. So I think that field will continue to deliver. There's so much out in the biosphere that can do fantastic things, and we can use it for our purposes.
Yeah, that's really inspiring. So to see the next generation of techniques.
Yeah.
Yeah, but then you moved from Caltech, and did you go back to Uppsala?
Yeah, I went back, not to the very same lab, but to lab. Ulf Pedersen was a professor of medical genetics, and he was a true mentor, extremely helpful for me. I initially, at the time, there were no starting grants in Sweden, so there actually was, in the daily newspaper, there was a picture of an interview with all of us who had been abroad. There was a group of some 10 or so who received a Wallenberg grant to go abroad. And it was a very sorry lot, because we in the interview, we all looked very sad. We said that, well, we had a fantastic time out there, but now there's no money. We will not be able to do any research. So now almost all of us are successful professors or retired professors by now, but everybody did very well. But there was no good mechanism to get people going. Now there are lots of, maybe too many starting grants, so there are lots of opportunities for young people to get funded. Still, not everybody. The problem is that they are piled on top, so some get lots of these grants and many get nothing. But at the time there really was nothing. But then the Bayer Foundation wanted to fund genome research, and there was a trio of us homecoming students that were offered this grant. It was interesting also because the three of us, we were all among the pioneers of PCR. We had all been very close to PCR. The three were Svante Pääbo, who later became very famous, Ulf Gyllensten, and myself. Gyllensten actually went to CITES, the company that developed PCR. Svante Pääbo intimately collaborated with them and was an early user and built his career on that. And I was the first to get the PCR machine outside of CITES. So we all had a very advantageous position, even for America. I don't think there are postdocs or PhD students that were in such a close contact with PCR outside of CITES. There were talented people in CITES that used to. Anyway, so we all got an offer to starting grant from the Bayer Foundation. Svante was recruited to Munich and never came back to Sweden, unfortunately, but did very well. Mm-hmm. But Gyllensten and I have been working along, and in a way, all three of us have built on the PCR. Svante for archaeological purposes or paleontological, understand the basis of human. Ulf Gyllensten has been very productive in advanced molecular genetics, but also he was one of the very early users of the protein, the proteogenomics that has been a topic here. So he was one of the first to use the Olink technologies and the kind of things that people are now publishing. How you can tell the age of a person by measuring proteins, and you can tell the height, how protein patterns correlate with lots of things. He published a very impressive paper in 2014 on a relatively small scale, 1,000 individuals and just 92 proteins. But that has now been built upon by UK Biobank and others, and they can corroborate and extend that finding. The protein provides a lot of very valuable information.
In going back, the three of you were really able to continue working on innovative projects and also develop technologies.
I think I have a medical background, and I was put in charge of medical genetics for the medical training, and I think I tried to transform myself into a medical geneticist, but I never did anything useful. I didn't really have the mindset that I will become a technologist, but it was sort of, it just happened, it kept happening that I found myself doing more technology than really interesting biology. And so nothing much came out of the medical genetics work. However, we came upon some technologies that have proven useful for us and for others.
Yeah. Can you tell me a little bit about those?
Shortly after the invention of PCR, I came up with an idea which is not at all as powerful, but still useful mechanism, where we used two synthetic oligonucleotides and allowed them to hybridize next to each other. And if there's a perfect match, then you can join them by ligation. If there's a mismatch of even a single nucleotide, then you cannot join them. So it was an early genotyping technology, and it has been used a lot as a basic mechanism by others and also by us to some extent. Coming back to Sweden, we just modified that. So the two oligos, we designed them to just be the two ends of the same linear oligonucleotide. So you have the 5' and the 3' end of one longer oligo sit down next to each other on the target sequence. And if there's a perfect match, then you can join them, and that creates a DNA circle. But because duplex DNA, where it's hybridized, is a double helix, it means that this circle will be wound around its target. So we called it padlock probes because they sort of lock in place. You can't remove them without breaking phosphodiester bond if there's a long target. And I guess that was maybe what made me optimistic that this would be a very powerful technology. Later we found other ways of taking advantage of this mechanism. So if you have a circular molecule, then you can use particular polymerases to copy the circle, and it will reel out copy after copy of this long DNA, this little circle, and generate a long DNA strand via a mechanism called rolling circle amplification, which has been fundamental. So if you have a very specific circularization of padlock probes, then only when that happens will you get this rolling circle product. So you will not get the product without the specific detection. And the padlocks are very specific. So they, like PCR, they are specific enough to look through complex genomes, such as the human genome. As I said before, in any cell, you have 10 to the 10 nucleotides to look through. And that's a lot of nucleotides to look through. So you need to input a lot of specificity if you want to find the right one. PCR allows you to do that because you require two oligonucleotides of some 20 or so nucleotides to hybridize, and that's going to be rare that they do in the wrong place, so you mostly get the right product. Padlocks have a similar. They also have 20 nucleotide sequences at the ends that have to fit, and if they don't, they will not. So the world record right now, there's a Thermo Fisher product based on something we did a long time ago, where they type 220,000 points in the human genome. They had 220,000 probes of the padlock probes to type positions in the genome. So it's highly scalable, and that was the other key, that PCR can't be scaled very well. It's a powerful technology, but you can't use it for very large numbers, so at least not easily. But padlocks can be scaled to any number. So that's been one thread. Later, we realized that this trick of ligating things we could also apply in the world of proteins if we put the oligos on antibodies. So if two antibodies bind the same target protein, they will be brought in proximity, and that will allow oligos sitting on the antibodies to be joined by ligation. And that's technology we call the proximity ligation assay. Olink has now modified that, so they don't use a ligase, they use a polymerase, and they don't join the DNA, but they allow the DNA strands to copy each other. But it's both variants were covered by our patent. So the padlock for DNA analysis, then came proximity ligation and proximity extension for protein. But we also combined the two so that with a different design of the oligo that sit on the antibody, you can generate DNA circles if two antibodies bind in proximity, like the padlock circularization. So and that now allows you to get localized detection, because circles can be locally amplified by this rolling circle mechanism. So one of our companies now is using this mechanism, pairs of antibodies giving rise to circles that can be amplified to produce spot-like features. I think everybody is in agreement that if you want to look for with DNA pairs of oligos in PCR or pairs of oligos hybridizing in padlock or give you more specificity than the individual ones. With antibodies, it's the same since a very long time. Since the 60s, my colleague Leif Wide in Uppsala developed a sandwich immune assay that completely dominated protein assays. Again, it requires 2 antibodies to agree that, yes, this is the right protein. Mm-hmm. So you want this dual recognition. And what set Olink apart was that it was the first time you could have this dual recognition in a multiplexable format. That turned out. Because sandwich assay could not be, it's more like the PCR, it's possible for single targets or a handful of targets, but it's difficult to multiplex. But with the DNA readout, we could ignore any cross-reactivity and only look at the correct pair of products. So that's why Olink has been successful in scaling.
Yeah, so throughout this, you've had a close collaboration then with like the private sector maybe, or how did you come into, like, how did you move what you were working on out from the lab and into...
Sweden is a little unusual from other countries. Norway used to have the same policy as we still do, but you dropped that. In 1980, there was a Bayh-Dole Act in the US saying that inventions made in universities belong to the universities. Before then, it was not clear who really owned inventions. In companies, so it's the company that owns inventions that their staff makes. But in universities now, the similar situation in that it's the universities. And gradually, more and more countries have adopted this US practice. So when I was in the US, my invention there quickly went to Caltech, and Caltech licensed it to Applied Biosystem without really asking my permission, because I had no power over that. In Sweden, we still own our inventions. So I always tell people that if you're really clever and come up with good ideas, come to Sweden. It's a good place to be.
Yeah. So the scientist actually owns.
Yeah, we own the fruit of our labor, and economically it has some advantages, but it also gives you a great degree of freedom. You can do things that you think are good. You don't have to ask some office at your university for permission. You have to do that in Norway. And it makes things much more. You lost a lot of when you gave up this teacher's privilege principle. Much fewer patents were issued in Norway, and fewer startups. So it really was a change for the worse, I think. I think you should back up.
Again, it's important to preserve this creative freedom then.
The problem, I guess, is that if you're not at all thinking along the lines of commercializing your finding, then there's nobody tapping on your shoulder. At Caltech, there was a tech transfer office. They come by and say, have you invented something recently? And then they'll write the patent for you and find who can use it best. In Sweden, you're more left to your own devices, and the consequences probably are that some inventions go unnoticed, but people who are more interested in really seeing how far they can take their ideas, they have more freedom in Sweden. And I think that has had consequences. That was in the response to your question. So, coming back in the US, it was easy. I had a relation to Caltech, and Caltech to Applied Biosystem in my case. In Sweden, I struck up a relation with a local biotech company called Pharmacia Biotech at the time. But relatively soon, we found that we didn't have the same aims in life. So for a while, there was a relatively hostile period. And eventually, the relation I had came to an end. I had a contract for a while, but when that expired, then I set out on my own. And since then, we've been starting our own companies.
Yeah, so from your lab, it's been spinning out several companies, right?
Yes, right. And but even that became a little bit constricting after a while, because while Norwegian and other scientists, they don't own their ideas because it belongs to the university. My ideas came to belong to the company I started called Olink Bioscience. And companies have to be relatively narrowly focused, because there's always something that has a better chance of making money than anything else. So they put all their money on that. And early on, I was quite happy. What Olink did was it became a mechanism to spin out companies. So we spun out from Olink Bioscience a company called Halogenomics, which was acquired by Agilent, and a company called Q-linea, which is traded on the stock market. And then eventually in 2016, we spun out Olink Proteomics. So ideas from my lab could be commercialized by this company instead of by a university, which was, I think, a little better. But eventually I got a little sick of this also, because they automatically got all ideas from my lab. Unlike other Swedish scientists that completely controlled their ideas. And this deal ended in 2017. So I don't know how much detail you want, but Olink Bioscience had a relatively valuable, I thought, extremely valuable patent portfolio from my lab. And in 2010, somebody in a foreign company wanted to buy us. I was opposed to that, but my co-founders, who were my PhD students, they wanted some money. So understandably, they wanted to sell. That time, the company turned us down. But then they came back again in 2011, and I've told this story a long time. There was a Thursday around midsummer when we were told that on the Tuesday after, we will go into exclusivity. And that means that the company will do its due diligence, but there's no longer an opportunity for our company to talk to anybody else. We will have to completely work towards this company. So I had between Thursday and Tuesday, and on the Friday I got in touch with a guy who had just sold the company for a couple of billion crowns. So I asked him, would he want to? There was now a price tag, how much the foreign company was willing to pay after their due diligence. So I said, well, this is a price tag. So do you want to buy shares of those who want to sell their shares? It got very complicated because this guy, many people wanted his money. So there was also a colleague, a gynecologist, who was very interested in maternity care in Africa. So he brought this newly rich person to Tanzania on the Saturday after my meeting. And then I had until Tuesday to communicate via very poor 2011 email. Didn't work very well in Tanzania. While also trying to fight my PhD students who really wanted to sell to this foreign company at all cost. And it was a very stressful weekend. But eventually there was an email saying, okay, I'll buy the shares for those who want. But I didn't want, I wanted to keep the company. But my student dropped out at that point. The company then increased in value by a factor of 100. So it was not the best choice I made, but anyway, and then one of his requirements, this person who bought the shares, was that I would continue handing over IP until 2017. So from 2011, actually between 2004 and 2017, all my ideas went to this company. But since then, we can start new things.
Yeah.
I think we were supposed to talk about the sample facts, and it's one of several. So we've now spun out a company called Rarity, a company called Readily, a company called Sample Facts, and there's also a company called Secret Tech, which I have a smaller role in. Yeah,
and all of these are spinning out from your current lab?
Yeah.
Yeah.
Mm. Yeah, Secret Tech is a special case maybe because it was very much an idea of one of my students, and my colleague Masoud Kamali Maghadam's student, his own invention, but he did his work. The teacher's privilege that we have in Sweden that I mentioned is not only for professors, it's also for PhD students.
Right.
And patent law says that the patent goes to the person who had the idea, not the person who paid for it. So the boss doesn't necessarily have to be on the patent. I wasn't on the patent of this guy, but he was kind enough to give me a little part of the company, so I feel engaged in that also.
Yeah, so then you're also really bringing the next generation.
Yeah, well, I have a fantastic second generation. I don't take credit for them. I think they have found each other in my lab, but I have had extremely talented students, and only some of them have stayed in academia, but some of them have now become extremely powerful players in biotech in Sweden. Simon Fredriksson was the person behind Olink very much, so it was very much his ideas. He was the CEO of Olink, while the technology that is now commercialized by the company known as Olink today, which is a daughter company of the old Olink company. But so he really should have credit. Unfortunately, he sold his shares too early to gain. But he now has started other companies. And Ulf Landegren is another guy. He started this company. I mentioned a company that was acquired by Agilent, was called Halogenomics. It took him 2.5 years after he graduated to build that together with other PhD students to a point where they could sell it for a good sum of money to Agilent. And quickly thereafter started another company called Vanadis, which does prenatal testing, same group of former PhD students. And after 2 years, they sold it for 1 billion Swedish crowns to PerkinElmer. And he's now on to a third company called Moleculant. So they've been extremely efficient. Also, Mats Nilsson is another of my students. He is a little bit more like me, so he has stayed in academia, but he's been very effective also spinning out companies. Cartana is maybe the most famous one, which was acquired by 10x. So they now have a platform for multiplex transcription analysis in situ, which is built on Mats' work.
Yeah, because it's really inspiring to hear how you can, from as an academic scientist, also do so much innovation and also have so much that like moves into, I don't know what to say, like the rest of the world, move into society.
Yeah.
So since we're here also where there's a lot of focus on biobanks, what do you think about like the private-public collaboration and innovation perspective around biobanks?
Yeah, well, I think it's important for many reasons. Obviously, you need individuals to agree to provide their samples, but also the economy of biobanking depends on the public thinking it's worthwhile to do it. So it's important that we give something back. I mean, we need their sample and they should benefit somehow. I think there's a misconception often that the countries that focus on science will have a healthier population. I think that's only marginally true. They may have a wealthier population because you can commercialize things, but the health benefit goes to the whole world. They are not going to be restricted to the country where the invention was made or a product was developed. So the motivation for countries to focus on this is to help mankind and not their countrymen alone. So a mutually respectful relation between the public and academia is important, but academia can, again, another misconception I think is that you do your academic research and somebody gets healthier. And that there's no such connection. There's no jump from an academic paper to somebody being cured of a disease. We are lousy at developing products, having them approved for use. So we can't afford to do that in academia. So it has to be taken over to the next step. Academia is really good at exploring new things and having a broad knowledge, broader than maybe companies can master, even the bigger ones. So there's a very important role for academia. I sometimes give a talk that I call universities as machines to build companies. I mean, clearly we should do lots of things in universities, just maintain knowledge through the millennia, or these centuries. But we also are in a very good position to lay the foundation for companies in a way that's very difficult to do. Companies will always have something that is worth more to pursue. So it's more difficult, I think, to come up with completely new ideas and pursue them within companies. CETUS is a case in point, because CETUS was a cancer company developing cancer drugs, and then more or less by mistake, one of their employees, very low on the rung, not very high-ranking Kari Mullis, he came up with this idea of PCR. And PCR was developed very much against the wishes of the leadership of the company. So the because the company, they had an idea they would develop cancer therapies. What's this PCR? Who cares? So that was a lucky shot, and it happened in an industry. But often the development can happen, I think, in academia. We're very well positioned to develop new things, but we really need to work with companies. So in my case, I've been extremely much benefited by good relation to companies. Also, I have a tremendous respect for the expertise in companies, which we're lacking in academia. But we have a lot of knowledge and experience and capabilities in academia, which is lacking in industry. And unfortunately, both parties tend to look down on each other.
Right.
The academic perspective is that if you go into industry, you failed. You were not good enough to stay in academia and become a professor. And that's a complete misconception, people. But the industry, same. They think that, if you didn't have good enough ideas then you're stuck in academic and published papers, and who cares?
Yes, right.
Mutual disrespect, which is very unproductive.
Yes. Do you want to tell me about the company that is also represented here, right? Yeah. Which has some technology that could be really important for biobanks.
Most of our technologies have been in the area of molecular genetics, using tricks of synthetic DNA and enzymes to analyze nucleic acids on protein. However, I had a very talented PhD student, Jan Björkesten, who had worked at the company Olink Proteomics, and there somebody asked him to test if he could measure protein using their platform on dried blood spots, and he had did a project there, which was successful. So he came to my lab, and together we expanded on that, and we did a more thorough study. How well could we measure protein in the dried state compared to the wet state? What was the reproducibility on pairs of dried sample compared to pairs of wet samples, and things like that. And we got a hold of PKU samples, the newborn samples.
Yeah.
One of the speakers said that Denmark has the longest track record. I think Sweden has a 10-year older track record of PKU samples, but maybe I'm misinformed. But anyway, so we got a hold of, at the time, 40-year-old samples from Sweden and 30-year-old samples from Denmark, and then 30, 20, and so forth, taken at different intervals, and checked if we could measure proteins in these old spots. And it turned out that most protein were very stable. They look the same in people born 40 years ago from people born 30 years ago, and so forth. Some declined, so with storage, and they have really been stored. Denmark stores their samples at -24, and Sweden we store them at +4. So, but they hold up pretty well. A little bit better in Denmark.
Yeah.
So anyway, we felt that this was very powerful, could simplify sample collection, but you don't really want to look at blood samples, whole blood. You don't seldom measure protein in blood. So we looked for a possibility of separating cells and plasma. And there are some obvious ways. There are so-called Paul membranes that are used to filter material. So we thought of maybe having some hydraulic, you press something through a membrane, or but then the simplest possibility would be if we just put two membranes on top of one another, filtering on top of a suction membrane. But that didn't work. Everything stayed on the top membrane, so we felt we had to have a better contact. And then we took the two membranes and put them across a cylindrical surface like this glass. And then contact was very good, and you put a drop of blood and plasma went to the membrane. But it's difficult to sell cylindrical things with. Yeah. So then we came up with a simple idea of just having this little pop-up mechanism. You open a little booklet and two membranes are pressed against each other. One is ensures it's anchored at both ends, and the lower membrane, the suction membrane, as it were, a Wattmann filter, it's pressed against the other one. And that gave us sufficient contact that the drop of blood put on the top membrane would retain all the cells, but all the plasma goes to the bottom, or most of the plasma goes to the bottom. then you let it sit for a few seconds. This separation happens in seconds, but if after a minute or so they will have dried, and then you can close the booklet and stick it in an envelope and send it in.
Yeah.
And I felt that there was a great need, also because a lot of the biobanking has been predicated on the idea of genetic analysis. And with genetic analysis, once you have one sample from a person, and you know all there is to know genetically about that person, except tumors and somatic mutations otherwise. However, dynamic mutations like transcriptomes, proteomes, metabolomes, and so other things, they are very dynamic, and there you need repeated samples. And I think that has been a problem with the biobanking community, that for too long they've been thinking about just making sure that you have one sample from every person. But it's increasingly important to have many samples from each person. And then it has to be really simple to collect. Forcing people to go to a nurse may be too much to ask. And also the cost of storing can be prohibitive. So this dried samples, they solve many problems. It's very simple to collect. People can do it in their homes. You can send things in by mail. You don't have to have a cold chain to preserve them, and they can be stored in very low volume. For protein purposes, +4 is good enough. Maybe we're just looking into things now. If you're interested in other things like lipids, it may be that they oxidize and that they have to be preserved better. So maybe for some purposes, you want to keep them at a lower temperature.
Yeah, because you were also mentioning the missing omics, what we're not focusing enough on today.
Yes. Yeah, that was a plug for something we're about doing now. So using a mechanism very similar to the one that was commercialized by Olink, we're now measuring antibody responses. And I think, for a long time, people have realized that antibody specificity is very informative. In infectious medicine, you always take follow-on samples to see if the titer for antibodies against the bacterium or virus has increased as evidence that somebody had an infection. And in autoimmunity, we know that often the antibody responses predate the clinical symptoms by a long time. So we have automatically an opportunity for early diagnostics if we were just better at measuring our antibodies. But currently, most assays look at antibodies against one antigen in one sample at a time. There are some where you can look at a larger set, but not so many antigen in one sample at a time. But with our mechanism, this Olink-like mechanism, you can pool, you can look at lots of antigen specificities in large numbers of samples in one sequencing run and learn a lot. So it can become an omic technology. And I think of all the things, in all these discussions, I think there's a question, which are the most valuable markers, or which ones do we really need? I think there are things you can measure that are kind of meaningless, that don't tell you anything, or at least that remain a little undetermined how valuable they are. Protein comes in a category of extremely meaningful markers, in my view. I think they tell us a fantastic amount, surprising amount of who we are and how we're doing health-wise. But antibody, I think it's well established that it's highly informative, and it's a little surprising that we don't have omic technologies. So that's something we're working on.
Yeah.
And it goes well together with the dried samples, by the way.
That's going to be really interesting to follow for the future.
Yeah.
Yeah. So as we round up here, I also just want to hear how you feel about the conference. Yeah,
no, I think it's very interesting. I was a little confused. I think it started as a biobanking conference and then merged with a precision medicine conference. But the connection is very tight anyway, so it was very logical to combine them. Yeah, no, I think there's a great optimism, and people are working effectively together. There's been some complaints that we're not working together in the previous session, that we don't work well enough together. But the Nordic countries, I think, stand out internationally, that we have a long tradition of collecting samples, and it would be logical for us to work more intimately together, I think. often you're limited by the numbers. If you're looking at rare diseases, each of our countries may not have enough of them. So I think there are many good reasons why we should be working together, which has been a theme. Many people have touched upon that. Sweden may stand out in having this technology perspective because besides my own work, there are many other. The whole area of spatial transcriptomics and single-cell transcriptomics was to a great extent pioneered in Stockholm. So there's been this tools perspective coming out of Sweden, which can be combined with the more biologically oriented interest in the other Nordic countries. So I think we have things we can offer each other.
Yes, I fully agree. Well, thank you so much for your time. It's really an exciting story and important perspectives.
Thank you very much.